let's finish respiration. We're on page 143. I'm going to finish talking about that today. So we're only missing Jordan, huh?
It's a doctor.
I'm going to see who's late today. No!
We're not doctors. We got to get out of here. What movie? Doctor, 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 and doctor. We're not doctors. We got to get out of here. You don't know? It's called Spies Like Us. You need to watch Spies Like Us if you haven't seen it.
Computers can do that?
Hilarious. Enable that.
This is going to be cool.
Beavis and Butthead?
Yep.
Nailed it.
Beavis and Butthead.
Finally got one, and no one else got it.
Very nice. Okay, so
Good reading.
the book's kind of hard to follow on this because the book can't show moving things, so I like to do this on the board. So please use your book as a reference here. But here we have, remember we had glucose, the hexagonal molecule that entered the cell. And as soon as it entered the cell, do you remember what happened? Pyruvate. A process happened. Glycolysis. Very nice.
Doesn't that kind of pyruvate?
It does. It forms pyruvate.
I thought you asked what happens with glycolysis.
No, I said what process happens. So it's glycolysis. G. Good people can achieve. So glycolysis happens first. And the glucose gets torn apart. And whenever a large molecule gets torn apart, stuff flies off of it. Like protons and electrons. Woo! And where do they go?
Inside.
They're picked up by electron carriers like NAD. I'll take those electrons and protons, the NAD says. And the NAD will eventually go inside the mitochondrion into the electron transport chain, which we'll get to in a minute. And now we're not left with glucose anymore. What do we have, Emery?
Pyruvate.
Pyruvate. Emery knows. Do the rest of you know? This is called pyruvate, and that's what's left after glycolysis. Oh, and in the process, some energy came out, and we made some ATP too. Do y'all remember that? We made a couple ATPs down here in glycolysis. That's our review. You should already know this. Then, what happens to the pyruvate if there's oxygen present?
Acetyl-CoA.
Again, you're just saying names. You're saying nouns.
The
when I'm
preparatory
asking for verbs.
reaction.
What's that? The preparatory reaction.
The pyruvate
Yes.
goes into the mitochondria and then it hooks on.
Nice. The pyruvate goes into the mitochondrion, and there's some CoAs waiting, and the pyruvate hooks to the CoAs, and carbon comes off, and now we have acetyl-CoA. And this carbon exits the mitochondrion in what form?
CO2.
Carbon dioxide. That's the carbon dioxide, some of the carbon dioxide that you end up breathing out of your body. And in that process, when we tore apart this larger molecule, you know what happened?
Energy.
Not energy.
Electrons and protons.
Protons and electrons came off of the structure. And where do those go?
NAD.
NAD comes and picks them up. There happens to be NAD just sitting around in the matrix. And it picks up the electrons, and it picks up the protons. And now we have NADH, and that's just going to slide right over here to the electron transport chain we'll get to in just a minute.
So wait, you just described the citric acid.
No, I just described the preparatory reaction.
Okay, well I missed.
I can tell you about the citric acid.
whole- I think I missed the end of the prep reaction then.
Here's the prep reaction. Just look up here, EKF. Pyruvate attaches to CoA. This carbon comes off and goes away as carbon dioxide, and some protons and electrons come off and get picked up by NAD. And NAD carries the protons and electrons to the electron transport chain. How we doing? Savannah, you good? Okay. Jonathan, how are you? Good. Gotta check on the back row. Sarah, you good? Jessica?
So what did it respond to?
NAD is the electron carrier that happens. Now, what I was talking about yesterday when the class broke up was the next step, the citric acid cycle, where this acetyl-CoA is completely destroyed by enzymes that tear it up, and boo, boo, these pieces come all the way off, boo, boo, and it frees the CoAs up again. And whenever you tear apart a large molecule, you know what happens? Protons and electrons and energy often flies off. And so again, when we're tearing up this molecule in the citric acid cycle, protons and electrons are flying off of the structure and getting picked up. Some are picked up by NAD, and it goes over here. And some are picked up by a different electron carrier you haven't seen yet, called FAD. And we like that one because we're Frederica Academy. And FAD comes up, and as this thing's torn apart, electrons and protons fly off, and FAD will pick up a couple of the protons and a couple of the electrons and carry them over to the electron transport chain. So FAD is just like NAD. It's just another carrier of protons and electrons. Just shuttling them on over.
Don't they pick up hydrogen?
Yeah, hydrogen is the proton. This is a hydrogen ion, and that's the same thing as a proton. And what happens to all this carbon that was part of our acetyl molecule?
It uses out of the cell and goes into the lungs.
It floats out. In what form does it float away?
Yes.
CO2.
Carbon dioxide. That's more CO2. So you see, after the citric acid cycle, our glucose came in and our carbon dioxide left.
What are those big things?
Oh, this is for the regular bio class. They're chromosomes. That's something we'll do later. Now, we're not done yet. We still haven't made very much ATP. There was actually a little bit of it made here in the citric acid cycle. The citric acid cycle, when this thing was being completely torn apart, we also put together a couple more ATP there. So we made a couple ATP in glycolysis. I think we made 2 of them. And in the citric acid cycle, we made a couple more. So far we've made 4 ATP from 1 glucose molecule. But you know how much we're going to make after it's all said and done?
6.
I hadn't told y'all this yet?
Wait, what's the question?
36 to 38 ATP. We've made 4 ATP thus far, and we're going to end up with 36 to 38 ATP. Where does all the other ATP come from?
Electron transport.
The electron transport chain. And that's what I'm going to show you next. Are you ready for the electron transport chain?
Okay.
Hannah, are you ready for the electron transport chain?
I'm so ready.
That is awesome. I knew you were excited about it, but here it is, finally. Here's the electron transport chain. It's just like the one we studied in photosynthesis a few days ago. It's proteins on the membrane. And what happens is these NADHs and FADHs that we've built up through the breakdown of glucose. When we broke down the glucose, the protons, the electrons were all picked up and were carried over here to the electron transport chain, and the electrons are dropped off at the first protein here in the chain, and the electrons move down the chain, just like they did in photosynthesis. And of course, as we drop off these electrons, the protons fall off too, and now the NAD is free to go back and get more if it needs.
So there's an electron transport chain in mitochondria and glycolysis?
That's correct.
Or do they have the same proteins?
They're a little different. They're a little different, but they're pretty close. There's no photosystems, no. This doesn't need light. So once again, NAD comes up, drops off its electrons, drops off its proton. The electrons move down the chain. The electrons move down the chain. And just like in photosynthesis, as the electrons move down the chain, some of their energy is used to push hydrogen outside the membrane.
So the hydrogen goes out into the...
Yes, the hydrogen goes out of the matrix, that's the middle part here, to this area here, which also has a name. You know what it's called? Those of you who know mitochondrion well?
Christy.
Intermembrane space. This is called the intermembrane space.
Well, don't the ions go in during...
In photosynthesis, they went in, but in respiration, they go out. Photosynthesis and respiration are opposites.
What does the FAD do?
Good question. Jessica. The FAD also drop off electrons. But they drop them off at a further protein down the chain. They don't drop them off at the beginning. They have a different drop-off area. It's like the lower school drop-off area instead of the high school drop-off area. And they drop off their electrons and they drop off their protons too. So the FAD is basically just like the NAD, Jessica. It just drops off electrons at different spots.
It's kind of like photosystem 1 and 2.
Sort of, a little bit. But don't think there's any chlorophyll or light or anything over here. Just different proteins. And so, again, as the electrons move down, hydrogens get pumped out. And if this continues to happen, whoops, I dropped one. If this continues to happen, NAD dropping off electrons, electrons moving down the chain, hydrogen getting pumped out, or protons getting pumped out, what's going to happen if that continues? We're going to build something up. You know what it is?
H pluses. Water.
We build up a lot of hydrogens on this side of the membrane. And not many on this side of the membrane. They're all getting pumped out. What do we call that?
Active transport.
No? Chemiosmosis will happen. What do we call it when we have a lot of stuff on one side of a membrane and not many on another side of a membrane?
Hypertonic.
No?
Hydrotonic.
No?
Acidic.
No? It's a something gradient.
Concentration gradient.
We formed a concentration gradient here. A high concentration of ions on one side of the membrane and a low concentration on the other side of the membrane. That's a concentration gradient. The same thing happened in photosynthesis. We can also call this a proton gradient because these are protons. And here's the big part for the cell. What are those protons going to want to do if they're in high concentration here and low concentration here?
Go back in.
They're going to want to go back in, and there's only one way back in, baby. Can you guess? It's the same thing as in photosynthesis.
ATP synthase?
through this special protein called ATP synthase. And when a proton goes through the ATP synthase protein, guess what happens? Y'all should all be yelling this out at once.
This is the other thing.
You should be incredibly excited.
ATP is formed.
Glucose.
ADP and P,
Split up.
when a hydrogen goes through the ATP synthase, ADP and P are put together
ATP.
to make ATP. And that's where most of our ATP comes from. These hydrogens going back in through the ATP synthase and ADP and P coming together each time that happens.
So it's like.
So in the electron transport chain, we end up making about 34 ATP this way from every glucose molecule. Because every glucose molecule that got broken apart left NADHs and FADHs that drop off protons and electrons in this chain. And as the electrons move down the chain, hydrogen gets pumped out, it comes back in, and we make a heck of a bunch of ATP that way.
So it's not really the electron transport chain that makes anything. It's the hydrogens that get pumped out by the energy.
You're right. But they're pumped out by the proteins in the electron transport chain. And when they come back in, that's when the ATP gets made.
Because
So,
of the electrons.
because of the electrons, that's right. Yes, Anna.
Where do the electrons come from? Because there's not like.
These electrons, they came from glucose.
Do they just like? How do they get onto the chain from like the NAD?
The NAD, remember the NADs picked up the electrons as we were destroying that molecule, and then let me put some electrons on an NAD. So, like for instance, as this was breaking up, we formed NADH. Electrons came from that and got onto NAD. Are you still with me, Hannah? Yes? And then the NAD kind of just cruises over here and drops off the electrons. The NAD is just an electron taxi cab. It's an electron carrier. And it just moves over here, drops them off, and the electrons go down the chain. Yeah? Good? You happy? You like NAD, don't you? NAD just moves by diffusion. It's a molecule and it's bouncing around, and whenever it hits that, the electrons will jump off. Other questions? Come on. Are y'all serious? Y'all didn't even know what was going on here and you don't have any questions?
Oh, it pumps hydrogen through.
Hydrogens go out, they come back in, and ATP is made.
And it's just a cycle.
Happen on the other thing?
Yeah.
Is it the same thing?
No, it's older.
They're both chemiosmosis. They're both movement of hydrogens out and then come back in. That's chemiosmosis is coming back in, and that makes ATP.
But it's not the same thing.
It's happening in the mitochondrion here. It was happening in the thylakoids there. But it's still production of ATP in the same way.
Okay.
Yeah.
In photosynthesis, wasn't it the hydrogens going out of the cell?
Yes, it was opposite.
Yeah. That made ATP instead of going in.
Yes, but it's not going out of the cell.
I
It
mean,
was
that-
going out of the thylakoid.
Yeah, the thylakoid.
What's the purpose of a plant cell having a mitochondria?
To breathe?
Alright. Because plant cells need ATP too for their cells.
I thought that was created by the
The thylakoid made ATP so glucose could be put together.
Oh, okay.
And then the glucose is torn apart here and ATP is made that way.
Alright.
Yes?
So, is there oxygen used in this step?
I'm getting there. That's the big finish. Are you ready for the big finish? Did someone else have a question just about this before we get to the big finish? Take a deep breath before we get to the big finish. Because that's what breathing is. The big finish is breathing. Oxygen is needed. Didn't I say oxygen is necessary in the mitochondrion?
Yeah.
Here's where it's used. Here comes O2. These are two oxygens hooked together. O2. It comes into the mitochondrion. It goes all the way down here to the end of the electron transport chain in the mitochondrion. and it picks up these electrons. And when it picks up electrons, you know what else it picks up?
Hydrogen.
Protons. Bam. It'll pick up 2 of them. Bam. Oh wait, let me do this. Let me do it this way. It picks up electrons here. It picks up protons here. And you know what we have now? We don't have oxygen anymore. You know what we have?
Water.
We got water.
Wait, where did those
Oxygen comes in,
come from?
and at the end of the electron transport chain, it forms water. Let's watch again. Let's go. Let's rewind. Here's oxygen O2. EKF, you looking? Here's O2.
It just comes in from the cell.
It comes in from your this is why you breathe. You breathe, you get it into your lungs, it gets into your blood, and it goes to every cell. And then the oxygen comes into the cell, then it comes into the mitochondrion, and it goes right here. This is the only place in your body, in your cells, that oxygen is necessary. Oxygen comes in right here at the end of the mitochondrion, picks up these electrons that have been moving down the chain, picks them up. When it picks them up, it becomes negatively charged. So these hydrogens that come through the ATP synthase, they stick to it. And ATP gets made, and then they stick to this oxygen that has the electrons, and bam, we've got water. And so oxygen is the final electron acceptor at the end of the electron transport chain in the mitochondrion. Where does it get what?
It's the hydrogens.
The hydrogens, remember, were dropped off by the NADs, and they got pumped out, and they came back in to make ATP, and then they get picked up by water. Yes?
Once ATPs form, where do they go?
They diffuse out of the mitochondrion through special ATP channels, which I'm showing you right here. And they go out of the mitochondrion, and now they're in the cell. And they just diffuse around the cell until they hit something where they're needed, like the motor molecule pulling a vesicle. And then they'll break apart, and the ADP and P will float back into the mitochondrion and eventually back here, and then get put back together again. And that's what ATP does. It just cycles around cells, getting torn apart whenever it's used, releasing its energy, and then going back to the mitochondria to get put back together. Question? Dante, question?
No. Oh.
Yes, Isaac.
This oxygen's used in the ETC step, but is it also used in the glycolysis step?
No.
Well, isn't that what fermentation is? If you have oxygen, it occurs.
You
No,
don't have
you
enough
have
oxygen.
You don't have oxygen.
So check this out. Here's glucose, right? In glycolysis, it's broken into 2 pyruvates. Remember I said those pyruvates are poisonous? Now, if oxygen is present, they can go right into the mitochondrion and start being broken down. But if there's no oxygen, then there's nothing here at the end of the electron transport chain to accept the electrons. And so that backs the whole system up. And so pyruvates stop being torn apart in the mitochondrion. And then no new pyruvates will enter.
I see. So it makes.
So if you're not breathing, the pyruvates can't enter your mitochondrion because the whole thing's backed up. So they're stuck here outside the mitochondrion where they're poisonous. So the cell, in defense of itself, does fermentation and makes these things into products that aren't as toxic.
That makes sense. So it's
Smart
required,
cell.
But it's not really. It's required, but not directly. Oxygen is only needed directly in the ETC.
Oxygen's needed in the electron transport chain. That's the only reason you breathe. Good, you're yawning. That's perfect. That demonstrates my point exactly. To keep your brain awake, you had to yawn to get more oxygen in. Good job. Good job, Jessica.
else.
Emery, are you okay? You're like totally just like, I can't take any more of this. Sometimes when I get like that, I jump and then feel better. What do y'all think? Is this like totally too hard to understand?
Well, it makes sense.
Not that bad, but bad question.
This is the hardest thing in AP biology.
Are you serious?
This is it. It doesn't get any more
No,
difficult.
the other one was so hard. No.
Photosynthesis?
Yeah.
Come on, EKF.
You come on. Photosynthesis is just like the last
You're right. You're right. Photosynthesis is tough too.
Photosynthesis and the Calvin cycle are definitely the hardest.
Okay. Photosynthesis and respiration. Those are the hardest
This is so
ones.
easy. This is like 5 steps.
I like it. I like to hear
Well,
that.
the other one's like 100 steps.
Well,
Wait, so it doesn't get harder?
nope.
Wait, you're kidding.
If you can do photosynthesis and respiration, that's as hard as it gets.
They said chapter.
The immune system is
So our
pretty
grades should
tough.
go up from here.
What?
So our grades should go up from here.
We'll see.
Oh, God.
First it should go down.
Mine doesn't go.
Really?
Why do they do this? I can only get better.
Because this is something that happens in the cell, and we go up from the small things to the large things.
Now we're doing it.
Questions? Come on, questions about this? Yes, Isaac.
I was wondering about the difference between 36 or 38 ATP.
This is a good question.
What's the question?
They do a count of the numbers where all the ATP comes from. And the book says, well, it's either 36 or 38 ATP. Do you remember that there was some NADH? Let me build another NADH molecule. Do you remember there was some NADH made outside the mitochondrion in glycolysis?
Yeah, where'd that go?
Do y'all remember when glucose was first torn apart, we did make a couple NADHs? And then they had to go inside the mitochondrion to get to this electron transport chain. Moving those NADHs into the mitochondrion, that requires energy. That's active transport. And it uses an ATP for each NADH. So if you consider that we lost those 2 ATPs by actively transporting NADH in, we really only get 36 ATPs from each glucose. If you don't count those, which I don't know why you wouldn't, we get 38 from each glucose. So one glucose molecule yields about 36 or 38 ATPs every time a glucose molecule is broken down. They talk about some of that accounting of where the ATPs come from at the end of the chapter. It's not that important. You need to understand how the ATPs get made. And that's the protons out and then protons back in mechanism, which we call chemiosmosis. Production of ATP by chemiosmosis. And that's the way most of the ATP gets made. It's a slower process. Glycolysis can happen real fast, but the rest of this is a slower process. So if your cells need real quick energy, they'll just do glycolysis and fermentation. They won't worry about this. That's what happens if you're sprinting or something. But if you're running a marathon and you're not using your energy too fast, you'll make all this extra ATP, and you'll be able to run way, way farther because you're using aerobic respiration. You're taking in oxygen to help break down your food. This is of such ultra importance to living things, y'all. It's why you eat, to get the glucose, and why you breathe, to act as an electron acceptor at the end of the electron transport chain. You should no longer answer those questions the way you used to. If I ask you why you eat, don't say, 'cause I'm hungry. Don't say, I need it to live. If I ask you why you breathe, don't say to live. Those are terrible answers for an AP biology student. If I ask you why you eat, you say, my cells require glucose to make ATP. And if I ask you why you breathe, you say oxygen is the main electron acceptor at the end of the electron transport chain in the mitochondria. That's why you breathe. Hey, let me show you something else that's pretty cool. Do you see how we make water here? We take in the oxygen, and that makes water at the end of the electron transport chain. That water has a name. It's called the water of metabolism. Some of the food you eat ends up as water inside your body. There are animals out there that never drink in their entire lives. Desert insects and such, they never even see any water. They just eat things, and some of the water is produced through this process. And that's enough water to sustain them for their entire life.
Why isn't it enough to sustain us?
Because we need more water than just that.
But we have a lot more of this stuff than they do.
It's still not enough. We need even more. We dry out too easily. They have extra layers on their body to keep the water in, and they don't dry out. But we do. Every time you breathe, some water comes out. When you sweat, water comes out. You need more water than just the water metabolism can provide. Isn't that cool? Desert insects.
What's that noise? Up there.
Probably the fluorescent lights. So 8.5.
Where did I find my turn?
Okay, video footage of some of this stuff. Here's the trick. Do you just sort of know this, or do you really know it? If you really know it, you can follow these videos.
I think I got this. It's called the Krebs cycle.
The Krebs cycle is the citric acid cycle.
Okay.
Let's just watch this one. This is the citric acid cycle.
The next stage of cellular respiration involves the movement of pyruvate into the mitochondria, where it undergoes oxidation. Each pyruvate molecule is converted into a compound called
Alright, here we go. So this is after glycolysis. We have the two pyruvates. You ready?
where it undergoes oxidation. Each pyruvate molecule is converted into a compound called acetyl-CoA. In the process of pyruvate oxidation, electrons are transferred to NAD, producing NADH, and a carbon is lost, forming carbon dioxide. The next stage is the citric acid cycle, also called the Krebs cycle. Here, acetyl-CoA will bind with a starting compound called oxaloacetate, and through a series of enzymatic redox reactions, all carbons, hydrogens, and oxygens in pyruvate ultimately end up as carbon dioxide and water. The pathway is called a cycle because oxaloacetate is the starting and ending compound of the pathway. For every glucose that enters glycolysis, the cycle completes twice, once for each molecule of pyruvate that entered the mitochondria. During pyruvate oxidation and the citric acid cycle, a net of 8 NADH, 2 FADH2, 2 ATP, and 6 CO2 are produced for each glucose molecule. In order to understand how the majority of the energy is produced by aerobic respiration, we need to follow the NADH and FADH2 molecules to the next stage, the electron transport chain.
So all we did was we took the acetyl-CoAs and broke them down in this cycle that I told you you didn't have to memorize. And we made 6 NADHs and 2 FADHs and some ATP. And we lost 1, 2, 3, 4, 5, 6 carbon dioxides total. Now let's look at their electron transport chain video.
glucose is oxidized during glycolysis and the Krebs cycle, the coenzymes NAD+ and FAD are reduced to NADH plus H plus and FADH2. In the mitochondria, the electrons from NADH plus H plus are transferred to the electron carrier proteins, and the protons are transferred across the membrane. As the electrons move from cytochrome to cytochrome down the electron transport chain, more protons are carried across the membrane. Cytochrome C transfers electrons to the cytochrome C oxidase complex. Protons are also transferred to the outside of the membrane by the cytochrome C oxidase complex. The cytochrome oxidase complex then transfers electrons from cytochrome C to oxygen, the terminal electron acceptor, and water is formed as the product. The transfer of protons generates a proton motive force across the membrane of the mitochondrion. Since membranes are impermeable to ions, the protons that re-enter the matrix pass through special proton channel proteins called ATP synthase. The energy derived from the movement of these protons is used to synthesize ATP from ADP and phosphate. Formation of ATP by this mechanism is referred to as oxidative phosphorylation.
Yes, that's another word for it, oxidative phosphorylation. That's chemiosmosis at work. Oxidative phosphorylation is when you put the phosphate onto the ATP during this process.